Let's discuss some important experiments that helped in determining the nature of hereditary material. Four major experiments were there. First is Frederick Griffith's experiment involving mice and bacterium Diplococcus pneumoniae. Second is Avery, McLeod and McCarthy's experiment involving extracts of Diplococcus pneumoniae, enzymes that digest DNA and proteins. This was done in vitro. Both these experiments were related to transformation principle. Third is Hershey and Chase experiment on T2 bacteriophages. Bacteriophages are viruses that infect bacterium. Fourth and last is Frankel and Conrad's experiment on TMV or tobacco mosaic virus. This experiment led to the conclusion that RNA can also be the genetic material. The first experiment was done by Frederick Griffith using mice and bacteria Diplococcus pneumoniae. This experiment led to the discovery of transformation. The ability of Diplococcus pneumoniae depends on a polysaccharide capsule that protects it from phagocytic cells of the immune system. Diplococcus pneumoniae is of two types, the one with capsule and one without capsule. This one is virulent and causes disease. This one is non-virulent and does not cause any illness. The encapsulated one is also known as smooth type or S type of strain. The non-encapsulated is also known as R type or rough type of strain. When S type virulent strain is injected into the mice, disease is caused and ultimately the mice dies. When R type non-virulent strain is injected, there is no illness and thus the mice survives. When heat-killed S type strain is used, infectivity is lost and there is no disease. Thus the mice remains healthy. But when a mixture of heat-killed S type and non-virulent R type together are injected, strangely the disease occurs and the mice dies. From the dead mice, virulent S type strain was recovered. Thus there was a change or transformation in Diplococcus pneumoniae. The experiments done by Avery, McLeod and McCarthy was done to identify the molecules responsible for transformation. That is, the phenomena of transfer of characters from one strain to another. In this experiment, R-type bacterial cells were treated with DNA extract of S type. This resulted in R-type bacterial cells as well as a few S-type cells were also isolated. When this extract of S-type was treated with enzyme DNase that destroys DNA, the transforming ability of this extract was lost. But when this extract was treated with proteases, that is, the enzyme that destroys protein, no effect was there and the transforming ability of the extract remained. Thus, it was concluded that DNA was the genetic material and protein was not. Alfred Hershey and Martha Chase performed an experiment using virus T2 bacteriophage and host bacterium E. coli. This experiment showed that DNA is the sole genetic material in bacteriophages and all the information for viral reproduction is contained in the molecule of DNA. Bacteriophages are viruses that infect bacteria. Here is the generalized diagram of T2 bacteriophage in code. The DNA is present inside and the tail fibers. In this experiment, using the technique of radio labeling, DNA was labeled with isotope of phosphorus, that is P32, and protein was labeled with isotope of sulfur, S35. Two separate cases of E. coli were taken. In the first one, the bacterial cells were infected by phage having labeled DNA, and in the second one, bacteria was infected by phage having labeled protein code. When this mixture was agitated, the bacterial cells and the remaining viral particles separated. It was seen that in the first case, the bacterial cells were associated with labeled DNA, the viral particles were normal. In the second case, it was seen that bacterial cells were normal and the leftover viral particles were labeled with 35S protein code. Virus undergoes multiplication in these infected bacteria. Phage components are formed, which are assembled to form the phage progeny. The phage progeny was found to be associated with phosphorus and not sulfur. Thus, it was concluded that DNA is the genetic material. Lastly, Heinz Frankel Conrad performed an experiment using tobacco mosaic virus or TMV. TMV has no DNA. Rather, it consists of RNA as the genetic material. It is surrounded by a hollow cylinder made up of protein subunits. In this experiment, the viral particles were separated into RNA and proteins. When RNA alone was used, it was able to cause infection and lesions were found on the leaves of tobacco plant. While when protein was used, no infection was there and thus no characteristic lesions were found on the leaves of tobacco plant. In a related experiment performed by Frankel Conrad together with Singer, reconstitution of viral particles were done. In this experiment, HR virus was also used or Holmes Regras virus together with TMV. The constituent particles of both the viruses were separated into RNA and protein subunits. Using RNA of TMV and protein subunits of Holmes Regras virus, a new virus was created. This reconstituted virus was used to infect tobacco plant. When viral progeny was isolated from infected leaf, it was found to be exactly similar to the TMV, having the same protein code and RNA code. Thus, RNA had all the information for viral reproduction.